The next step in the process was to construct the gene trees. So only the neighbor joining tree worked. As I stated before, the other trees, maximum likelihood, Mr. Bayes, and the IQ tree, all failed. So next was the rate tests, and the rate tests were used to test whether or not the differences were large enough to be statistically different from chance. And this here is the results of my rate tests, and I used alligator as the main outgroup. For the true reconciliation, it's when a gene is compared to a consensus tree, and it estimates the inconsistency between genes and evolutionary history of different species. Next is the molecular clock. So I constructed the molecular clock using the gene tree that I previously created. And what it does is it estimates the rate of evolutionary change for the gene by estimating the slope of a linear regression. And so once I had this done, I compared my findings to the rest of the class. And for the results for this part, my slope was definitely an outlier compared to everybody else in the class, as it was at the very bottom, at 19 out of 19 compared to the rest of the class. So this information helped me to conclude whether my gene was essential or not. In conclusion, from all the information that I gathered, I can conclude that ABCA7 is likely not an essential gene, which means variation in these genes are likely to not be harmful to the organism bearing these variants. And it's an unessential gene, so that means it is not conserved and it is also fast evolving. So Alzheimer's can occur without the ABCA7 risk variant. Because of this, I am led to believe that the gene ABCA7 is not important in the phenotype differences. And here's my works cited. Thank you.